1 bag of lentils, a blender, 5 minutes. A peer-reviewed study published in 2025 found that a lentil-based kitchen preparation outperformed a $30 commercial rooting product across nearly every measured variable: plant height, root length, leaf count, fresh weight, root dry weight, and female flower production. Not by a small margin, by double in most categories. That study is indexed in an international scientific database. It has not been retracted. The global rooting hormone market was worth $1.21 billion in 2024. It is growing. The lentils cost less than $1. That gap between what is on the shelf and what is in your kitchen is not an accident of scientific ignorance. The molecule has been documented since 1880. The chemistry has been confirmed for nearly a century. The alternative has been used by farmers across Latin America for generations. Here is the question this video is investigating. Why does a $1.2 billion market exist to sell you something that science confirmed you already have for free? That is the investigation. If you want investigations like this one, documented, sourced, and kept off the shelf, subscribe now. There is a lot more coming. The lentil rooting technique is not new. Farmers across Chile, Spain, and Mexico have rooted grapevines, olive trees, and rosemary cuttings with sprouted lentil water for generations. The technique is documented in Spanish-language agricultural literature, community garden guides, and permaculture handbooks across Latin America. Search enrizante de lentillas, lentil rooting agent, on any Spanish-language forum, and you find thousands of detailed posts, most of them attributed to knowledge passed from parents and grandparents. Search lentil rooting hormone in English. Until very recently, you found almost nothing. That asymmetry has a precise institutional origin. In 1945, the same year 2004-D became the first synthetic herbicide sold commercially in the United States. The infrastructure for synthetic plant growth regulators was being established in the same post-war chemical conversion. Factories that had produced wartime compounds were pivoting to agricultural chemistry. The USDA Agricultural Extension Network, the system that advised American farmers and gardeners on every aspect of their practice, became the distribution channel for the new synthetic inputs. By the early 1950s, USDA extension bulletins across the country were quietly reclassifying traditional botanical knowledge in the same language used to dismiss cover cropping and minimal tillage. Natural preparations were labeled variable, unreliable, and not recommended for modern propagation practice. The vocabulary was not that these methods were wrong, it was that they were imprecise. In the framing of post-war agricultural modernization, imprecise meant inferior. That reclassification had a direct commercial beneficiary. Every gardener steered away from a kitchen preparation and toward a standardized synthetic product was a recurring customer. The irony that the botanical extension literature will not acknowledge. The molecule being sold in those synthetic products, Auxin, was isolated for the first time not in a laboratory, but in biological materials. It was first isolated from human urine in 1934, then found in yeast, then in fungi, then confirmed as universal across all flowering plant species on Earth. The molecule is not a product of industrial chemistry. It is produced continuously by germinating seeds, by plant roots, and by willow trees that have been growing on river banks for millions of years. The chemistry was known. The natural sources were available. The decision to sell the synthetic version while labeling the natural preparation as primitive was institutional, not scientific. The science now being published in peer-reviewed journals says the natural preparation works better. The extension offices still do not say it out loud. Charles Darwin detected auxin in 1880 without knowing what it was. Working with grass seedlings bending toward light, Darwin and his son Francis designed experiments covering different sections of the seedling with opaque caps. Their finding: something produced at the tip of the seedling traveled downward and caused one side to elongate faster than the other. He called it an influence. He could not isolate it. He only knew it existed, that it moved through the plant, and that it controlled growth in ways no one had documented before. 46 years later, in 1926, a Dutch graduate student named Fritz Went captured Darwin's invisible influence in a block of gelatin. He placed cut seedling tips onto agar, waited, then placed those agar blocks onto decapitated seedlings. The seedlings resumed growing. The substance that transferred growth was later identified as indole 3-acetic acid, the same molecule being sold today in every bottle on the garden center shelf. In 1934, the molecule was first isolated not from a plant, but from human urine. It was then found in yeast in fungi, and eventually confirmed as universal across all flowering plant species. Not one species tested in 100 years of plant biology research has been found without auxin. It is synthesized from tryptophan, the same amino acid found in food, through a biochemical pathway so ancient it predates the evolution of flowering plants by hundreds of millions of years. When any seed germinates, its first and most urgent biological task is to grow a root. To accomplish this, the seed floods itself with auxin. Hormonal concentration peaks exactly at the moment of sprouting, when tiny white root tips first emerge from the seed coat. That is the moment the seed is producing the maximum concentration of the same hormone that commercial companies synthesize in factories. Lentils are ideal for extraction because they germinate fast, 2 to 4 days at room temperature, produce minimal fermentation, are inexpensive and globally available, and release auxins readily into surrounding water without requiring specialized equipment. The preparation that the 2025 peer-reviewed study tested is documented and replicable. Soak 100 grams of dry, whole, uncooked lentils in 400 milliliters of water for 8 to 12 hours. Drain but save the soaking water. It already contains dissolved auxins. Cover the damp lentils with a cloth. Keep them moist twice daily, and wait 2 to 4 days until small white root tips emerge. That moment of peak sprouting is the extraction point. Transfer the sprouted lentils and soaking water to a blender with 1 cup of fresh water. Blend until smooth, strain through cheesecloth. Dilute 1 part concentrate to 10 parts water before use. That ratio is not optional. Auxin follows a biphasic dose-response curve where low concentrations promote root growth and high concentrations inhibit it. Willow water provides something different and complementary. Willow trees are among the most auxin-rich plants on Earth. The same compound sold in commercial rooting products, indole butyric acid, drips freely from willow twigs soaked in warm water overnight. Indigenous communities understood this without naming the chemistry. The Cherokee, the Ojibwe, and the Iroquois used willow preparations for centuries. An Egyptian medical text from approximately 1500 BC prescribed willow preparations for wound healing and growth. When indigenous peoples pushed willow stakes into riverbanks for erosion control and watched roots emerge within days, they were producing functional auxin solutions without knowing the biochemistry. But willow water adds something lentil concentrate cannot: salicylic acid. Salicylic acid, the compound Bayer converted into aspirin in 1897, triggers systemic acquired resistance in plant tissue. When a cutting absorbs salicylic acid, it activates immune pathways that fight bacterial and fungal infection. The primary killer of plant cuttings is not hormone deficiency. It is fungal rot at the cut site. Willow water addresses both problems simultaneously: the hormonal signal to grow roots, and the immune activation to survive long enough for those roots to develop. No commercial rooting product combines both functions at the molecular level. The combination technique, willow water as the blending liquid for sprouted lentils, costs under $1 per application. Equivalent commercial products cost $30 to $50 and are less complete. A 2025 study published in Horticulturae tested a lentil-based biostimulant against a commercial synthetic rooting agent on Italian zucchini plants. 5 treatment groups. Multiple concentrations of the lentil preparation tested alongside the commercial product, and a water-only control. Statistical significance assessed by Fisher's least significant difference test. The results at the highest lentil concentration compared to untreated controls. Plant height up 166%, root length up 165%, leaf count up 113%, fresh aerial weight up 169%, root dry weight up 116%, female flower production up 177%. Compared directly to the commercial synthetic product, the lentil treatment exceeded the chemical agent across nearly every variable. Plant height 13% higher, leaf count 124% higher, fresh aerial weight 103% higher, root dry weight 117% higher, female flower production 160% higher. The researchers' conclusion: lentil biostimulants could serve as an ecological alternative to conventional rooting agents. At the Royal Botanic Garden Edinburgh, one of the most respected botanical institutions in the world, a separate study tested natural Auxin preparations derived from willow and lentils against synthetic indole-butyric acid on Saxagathia conspicua, a threatened conifer native to Chilean rainforests. The study confirmed detectable Auxin levels in the homemade preparations and demonstrated genuine rooting potential. The natural preparations worked. The chemistry was real. The study was small-scale. A large-scale controlled replication has not followed. These studies exist. They are indexed and retrievable. They are not controversial within their methodology. They are simply not the studies cited in the marketing materials for products that cost $30 a bottle. The honest accounting of what the evidence establishes and does not establish. The lentil preparation works in documented trials. The chemistry is confirmed. The comparison to commercial products in controlled conditions shows equivalent or superior performance. What has not been established is a large-scale, long-duration, independently funded trial across multiple crops, soil types, and growing seasons that would generate the evidence base required for agricultural extension endorsement. That trial has not been run. The funding to run it has not appeared. The mechanism is not disputed. Auxin's role in root initiation is established across decades of peer-reviewed plant biology. The University of California Botanical Garden has documented how indole-3-acetic acid activates specific genes that cause ordinary stem cells to differentiate into root tissue. Electrotropism, roots growing toward electrical and chemical gradients, is first-semester plant physiology. The presence of biologically active auxin in germinating lentil seeds is confirmed by the 2025 study and prior literature. None of this is contested. What is disputed is whether natural preparations can be standardized reliably enough for commercial and research endorsement. The standardization argument is real and legitimate at industrial scale. A synthetic commercial product delivers precisely 3,000 parts per million of indole-butyric acid every time the bottle is opened. Commercial nurseries propagating tens of thousands of cuttings simultaneously require that consistency. The argument is valid for industrial propagation. The argument is not valid for home gardeners propagating a dozen cuttings in a backyard. A homemade preparation that delivers somewhere between 200 and 2,000 parts per million is adequate for the vast majority of home propagation tasks. The standardization argument was built for commercial nursery scale and applied wholesale to domestic use, in extension bulletins, in university horticulture courses, in garden center packaging, to justify a price point that domestic users do not actually require. The pattern has a structural explanation. The global plant hormone market generates $4.5 billion in annual revenue. The rooting hormone submarket alone reached $1.21 billion in 2024. The margins on synthetic auxin products are substantial. The active ingredient costs pennies per bottle to synthesize at industrial scale, and the formulation is protected by branding. Rather than a patent on the molecule itself, auxin is not patentable. What is sold is the branded delivery format, the precise concentration, and the consumer's belief that they cannot accomplish root propagation without it. The British Electroculture Committee, established in 1918, staffed by fellows of the Royal Society, spent nearly 2 decades documenting electromagnetic agricultural methods before its reports were sealed in 1936. The same extension infrastructure that buried that research was simultaneously building the framework for synthetic plant growth regulators. Academic citations for traditional botanical propagation methods dropped through the post-war decades precisely as the synthetic plant hormone market was being established. No internal memo has surfaced ordering the suppression of lentil rooting knowledge. The record shows something more structural, an institutional framework that consistently rewarded standardized synthetic inputs over traditional preparations, that classified the latter as primitive and variable, and that aligned research funding with the inputs that required annual purchase. Auxin is not patentable. A lentil solution cannot be branded or sold seasonally. A technique that anyone can execute with a $80.80 does not generate recurring revenue. The research into natural auxin preparations that would establish them as standard practice for home gardeners has not been funded, not because the results are implausible. The 2025 study establishes they are not, but because there is no financial structure to support funding an investigation whose positive result would sell nothing. Here is the honest accounting. The lentil technique works. The 2025 peer-reviewed study confirmed it in controlled conditions, compared it against a commercial product, and documented the results across 6 measurable variables. The chemistry is sound. The molecular mechanism is established. The preparation is documented and replicable. What has not been established? A large-scale, multi-crop, multi-season, independently funded controlled trial that would generate the evidence base required for extension endorsement, regulatory recognition, or inclusion in standard horticultural education. That trial has not been run. The institutions that fund agricultural research at scale are structurally aligned with the inputs this technique would replace. The funding that would confirm what the 2025 study suggests has not materialized, for the same structural reasons that the copper pyramid has no funded trial, that Electroculture research citations collapsed in 1936, and that the weed suppression properties of cereal rye were confirmed in peer-reviewed journals and never reached an extension bulletin. The molecule that Charles Darwin detected in 1880 is synthesized by every germinating lentil in every kitchen pantry in the world. The knowledge to extract it was documented in peer-reviewed literature. It has been used by farmers across Latin America for generations. It outperformed a $30 commercial product in a 2025 controlled trial. The rooting hormone shelf at your garden center exists, not because the alternative is inaccessible. It exists because the knowledge of the alternative was reclassified as primitive during the same institutional window, when synthetic inputs were being established as modern, and because the research that would update that classification has no financial sponsor. That question, who funds the trial that would confirm what the study already suggests, is still open. Here is what I need from you right now. Try the lentil technique this season. Document your results with a control cutting from the same plant. One word in the comments. What plant are you going to propagate first? I track every answer. And if this changed how you think about what that $30 bottle actually contains, share this with one gardener you know. The best way to return knowledge to the people it was taken from is to pass it from one garden to the next. Subscribe if you want to keep following this investigation. The next episode continues the same thread. There is a lot more buried out there, and we are just getting started.